Everybody please be seated. We are going to commence our second technical session. Everybody please seated. Welcome back everybody. Moving on to the next technical session, it's my privilege to welcome on stage Professor Nirmal Kumar Sarkar, who will be chairing this particular session. Sir, to the stage.
We are going to start the day's second scientific session. There will be a single lecture in this session, in this room, because a concurrent session, parallel session will be held upstairs, that is poster presentation session. But here we will have only one lecture by an eminent professor cum scientist, Professor Arnab Sen from North Bengal University. Before he starts talking, it's imperative that I shall be introducing the speaker formally. Professor Arnab Sen is Professor cum Coordinator of DBT Bioinformatics Facility Center of North Bengal University. He is the founder of the center and also the founder of Genome Center in North Bengal University. Professor Sen did his master's in botany with specialization in cytogenetics from North Bengal University and then earned his PhD degree on electron microscopy and molecular biology of the bacterium Frankia from Northeastern Hill University of Meghalaya. Professor Sen gained commendable postdoctoral research experience from University of New Hampshire, USA, and University of Lyon of France. Subsequently, he has mastered a vast arena of research which embraces cytogenetics, metagenomics, transgenics, micropropagation, molecular marker studies, nanobiotechnology, bioinformatics, drug designing, and network pharmacology. Professor Sen happens to be the founder of Bioinformatics Facility Center and Bishwabangla Genome Center in North Bengal University. In recognition of his brilliant research career, Professor Sen has been honored as Fellow of the Linnean Society London, Fellow of the National Academy of Biological Sciences, and Fellow of Indian Association of Angiosperm Taxonomy. He is a member or life member of different learned societies and statutory bodies of India and abroad. For instance, Society for General Microbiology, UK, American Society for Microbiology, International Society for Computational Biology, USA, International Association of Computer Science and Information Technology, Singapore, etc. Professor Sen has been honored with different awards like DST Fast Track Young Scientist Award, DBT Overseas Award, DBT Crest Award, and so on. Professor Sen acts as editor of Indian Journal of Microbiology, published by the Springer, NBU Journal of Plant Sciences, and Indian Journal of Biotechnology, published by the CSIR. Professor Sen acts as a reviewer of scientific papers of a number of Indian and foreign journals. Professor Sen delivers invited lectures as a resource person in different national and international conferences. One such noteworthy lecture was the Constance Ledward Rollins Distinguished Lecture at Department of Microbiology in University of New Hampshire of USA in 2007. Professor Sen has already supervised more than 20 PhD theses and has been continuing with several more. He has around 200 peer-reviewed research publications to his credit. He created a database for North Bengal bamboos and another database for Mycobacterium secretome analysis. He has also authored 2 books on useful plants and useful microbes and several book chapters as well. What I personally find to be praiseworthy about Professor Sen is his modesty as a scientist. He hardly shouts about himself. নিজের সম্বন্ধে প্রচার একটু কমই করেন উনি. Rather, he speaks in plural number. For instance, he hardly says, I have done this, I am doing this, I shall be doing that, etc. Instead, he says, the Bioinformatics Facility Center of North Bengal University is exploring the Frankia genome. The Bioinformatics Facility Center has several publications and so on. Hard work and since modesty together will definitely bring him long-lasting success. At least I believe so. With these few words, introductory words about Professor Sen, I cordially welcome him onto the dais and to make his valuable deliberations. About 40 to 45 minutes, so that it can be completed in time.
Well, thank you, Professor, for telling so many good words about me, though I don't deserve them. And also, I would like to thank the Midnapur City College for inviting me from all the way from North Bengal to deliver this lecture. Also, I was given a task to see the posters and evaluate that I have recently done just few minutes back. So, what I am going to tell you today is the in silico inquest of fight against recent viral outbreaks. Actually, viral disease in our childhood was not that popular, that common. That time mainly the bacterial disease was predominant, the malaria, malaria, this and that and that and that. But viral diseases was not much, except polio and other things, were not much heard. But in recent days, it has been found that the viral disease has taken a center stage. Virtually every year, a new viral diseases are coming up in new avatar. Sometimes it is MERS, sometimes it is SARS, sometimes it is swine flu, sometimes it is bird flu, and sometimes it is legendary COVID. So there is a serious need to fight against these viral diseases. And why these viral diseases are coming up? Actually, I have an idea, I have a theory in this case, and I have discussed with my students and colleagues regarding this, that see, bacterial disease we have almost conquered with the help of antibiotics. There are several antibiotics present there for various bacterial diseases. So the human needs something to die. So that's why, since the bacterial diseases are not there, virus has taken the center stage of these things. Now we have to fight against those viral diseases. But the worst thing is that, till date, there is very less medicines available, allopathic medicines, drugs, tablets available against viral diseases. We have seen when the COVID broke out, we found that there is virtually no disease and we do not have any clue to manage also. And then that time for the survival, what people have done and what not. they have taken malarial drugs, they have taken chloroquine, taken সব কিছু they have studied. So this story started when the COVID broke out in 2019 and 2020 early. That time we found there is a devastating viral disease that broke out and thousands of people are dying. So in our laboratory at North Bengal University, we sat with my students, our students, and we seriously discussed that how we can contribute in the fight against COVID. So what we thought at that time was that the North Bengal is usually a backward place. This place is usually not like Calcutta, that all the medicines are available, everything, particularly in the remote village of the hilly regions of North Bengal. I mean, going to a doctor, it takes a lot of time to see a doctor. But then how they can cope up with this type particular disease, because this COVID is also slowly grabbing those North Bengal regions, particularly the hilly North Bengal, though devastation was not that much. So I thought, and then we thought that maybe we can use whatever available resources are there in the North Bengal region. So what we have started, that since I am a man of botany, biology, we thought that there are, and North Bengal there is no dearth of medicinal plants. So we thought that those plants should be exploited for the management of COVID. So that was the story. But before going to that, I was told that a lot of undergraduate students will also be there. So I have thought that since this work is on in silico, that is bioinformatic part is there, and the virals also there. So I'll start with the bioinformatics or these things in silico biology, and then we'll go to directly, slowly to our main topic that I am going to talk. Now, what is bioinformatics? If I'm asked to define what is bioinformatics, bioinformatics is virtually, it's a marriage between the biology and the computational science. That means for bioinformatics, for the first time, two completely different disciplines. One is from the biological point of view, another is from the physical point of view. Physical science and biological science coming together to form a discipline. That's why it is calling a marriage between the biology and computer. But the story started long back. Probably you know the Human Genome Project. Human Genome Project is there in 1993, and last decade of the last century. US Department of Health took a giant step in sequencing each and every letter, 3 billion all letters of human genome, not just once but 9 to 10 times. So that time scientists have anticipated that with the end of this Human Genome Project, huge amount of data, ocean of data will be generated, and it will be virtually impossible for the human being to analyze this data until and unless we have a separate discipline for them, and until and unless we have a specialized software, specialized hardware, and specialized personnel for them. Considering this, a new discipline has been created which is called as bioinformatics or computational biology. And this started, and this computational biology and bioinformatics have done a havoc. In fact, you may have heard that only 1 week back or so, our Prime Minister has given hundreds of human genomes from the Indian subcontinent to the scientists for analysis. So Human Genome Project is still going on. In India also there are hundreds of human genome projects are going on these days. Now, how bioinformatics actually started, why I am saying that bioinformatics is not started with the starting of the Human Genome Project, but its concept was much better previously. Probably you know all these 3-4 scientists. Students must be knowing. His name is Francis Crick, Watson, the legendary two scientists, Rosalind Franklin, and Maurice Wilkins. These 4 scientists. What is common between these 4 scientists? These four scientists are responsible for the discovery of the DNA double helix. Out of these 4 scientists, Rosalind Franklin was a radiobiologist. He was working on the X-ray crystallograph of the human DNA structure, DNA X-ray crystal structure, and she has taken this photograph of DNA. Excellent photograph, and this is one of the all-time best photograph of human DNA crystallographic photograph. Now, when Watson and Crick have seen this photograph, they have an idea that the DNA, previously it was thought that probably the sugar phosphate backbone is in the center and the nitrogenous bases are bulging out from that. But for the first time, Watson and Crick thought that instead of one sugar phosphate backbone, there are 2 sugar phosphate backbone and they are running antiparallel. And the nitrogenous bases are virtually inside rather than outside. So that is the discovery of the DNA. But then all started with this, the analysis of this photograph. So this is also the starting of the computational biology or the theoretical biology. Because Watson didn't go to the laboratory, wet lab. They just analyzed the data because they were actually men of physics. But when they have done these things, when analyzes this crystallograph, they found the structure of DNA. So the concept of computational biology or theoretical biology or bioinformatics started from there itself. Now, what is the definition of bioinformatics? The collection, classification, storage and analysis of biochemical and biological information using computers, especially applied to molecular genetics and genomics. This is the dictionary of bioinformatics as per the Webster dictionary. The Webster dictionary is one of the best dictionaries of the world. Now, ultimate goal is to enable the discovery of new biological insight. Actually, what happened, what we thought previously that we do not know, but bioinformatics will tell us that that can happen. That can happen. Say for instance, I will come to that point. But what is the basic difference between the bioinformatics and the traditional biology? This cartoon shows that traditional biology is probably the fishing with a net, fishing with a cane. You can fish at one fish at a time, but with the fishing with a net, you can catch a lot of fishes at a time. So bioinformatics is like fishing with a net, and traditional biology is fishing with a cane. Now, what are the perspective of bioinformatics? First perspective is the cell. The cells are the building blocks of the life. So, understanding of the cell is very, very important. And you don't understand even the bacterial cell. When I have started around 25, 30 years back on bacterial cell, Sarah just told that on Frankia we have done the work. So we thought that maybe it will take 2-3 years to analyze all the bacterial Frankia cells. But after 30 years, I must confess that I do not know the 1% of the Frankia cell. It is so fast, so understanding of the Frankia cell, just one bacteria, is virtually impossible to do it in one lifetime, I am telling you. So you can understand that how complex is this. It looks like a very small, tiny cell, doesn't look, you cannot see it in naked eye, but the complexity and the intelligence of this particular cell is enormous. If you analyze this cell, then you understand that how they can function and how they can manage all the problems of the world with that only one tiny cells. So understanding of the cell is the first perspective of bioinformatics. Second perspective is the organism. Organism may be a unicellular organism or multicellular organism, but it is organism. But when there is organism, there is organogenesis, and understanding of this organogenesis is also very important in case of and the second perspective of bioinformatics. And the third perspective is that when the organisms are coming up, organisms are coming together, because no organism can survive singularly, alone. They join together to form a community, that is population. And population is not a static body. Population evolves. Always population evolves. Human population evolves, other population evolves. This way, from an ape, human has come, and then in future, a superhuman will be coming in future. So understanding of these things, but the basic part of the perspective of bioinformatics, that is the cell, the organism, and the tree of life. Now, let's come to the genome sequencing, because I have done some work on genome. There are various genome sequences are there. Already human genome, I told you about human genome. There are vertebrate genome, plant genome, microbial genome. There are hundreds. So this is an old, actually old slide. Hundreds of, now then more than 1 lakh bacterial genomes are now available in the public domain. And these are the pictorial parts of the things. Bacterial genomes and various other genomes that has already been sequenced. Now coming to that point, the 2007 I went to USA. Then we wrote a project along with Professor Luis Tiza to sequence 5 Frankia genomes. And that was the first Frankia genome sequence, serious Frankia genome sequencing. And the project has been written in US Department of Energy, and they have sanctioned it. So we have sequenced 5 Frankia genomes. After that, a lot of Frankia genomes we have sequenced, and these are 2 scientists who are pioneer in doing the Frankia genome sequencing, and particularly Professor Kelly Thomas. He has established a laboratory that is called as Hoover Genome Center, Hoover Center for Human Genome Studies. When I visited this center in 2007, I liked it very much. And then I thought that maybe, if possible, then we can also establish a similar center at North Bengal University. And after that, I tried, and we all together, all our students and colleagues, they tried hard. And then finally, we could establish a center at North Bengal University that is called as Vishwa Bangla Genome Center, that we have done. Since we have given the name, because the name is very pet name of our beloved Chief Minister, so we thought that maybe we can use this name, and instantly this particular genome center has become very popular, probably because of the name. So that we have done, and we have already now the building is also found, and this is working. Now in this all these days, we have done several Frankia genome sequences other than Frankia 5 genome sequences we have done, then Prosorella, Nocardia, Streptomyces, Rhizobium, all these genome sequencing we have done, and we have established a genome sequencing laboratory at North Bengal University. In fact, I have a lot of collaborators, in fact, with this genome center, and collaboration from France, collaboration from USA, collaboration from Portugal, collaboration from Tunisia. All my collaborators, they are sending data to our center, and our center, they analyze these things. Our center is mainly analyzes the thing, and we publish paper together. And a lot of good work has been done through this. Some of the papers. Now I am coming to the main topic, this how to fight against viral diseases. But before that, I should talk a little bit about computer-aided drug designing. Why computer is common in drug? Because, you know, the thing is, without computer also you can analyze the drug. You can discover drug. But it will take 20 years, 30 years, 40 years. Because drug discovery is not an easy task. And today, particularly today, the civilization doesn't have so much of time to give to discover a drug. Because you have seen, I have already told, that every new virus are coming in, new diseases are coming in new avatar, new ways, new forms. So you have to always fight against those diseases and to discover drug against those diseases in a reasonable time, within a few months. Say for instance, you have seen when COVID broke out, the vaccine has been prepared in only few months. But if it takes 20 years, 90% of the population would have died of COVID. It didn't die because the vaccine has been discovered. Also in India, also vaccine has been discovered in a reasonable time, within few months. So that is the thing. So the computer helps greatly in discovering the drugs. So that is called as computer-aided drug designing, or CAD. I am not going in detail of CAD because there are a lot of things to be talked. Now let's talk about the various ways of diseases. Just now I was there in the poster session, that one girl has told me that tuberculosis is a pandemic, but that is wrong. Tuberculosis is not pandemic. There are definitions, perfect definition of various mode of diseases, various expression of diseases. So we need to understand all these things properly. First thing is the pandemic. Pandemic means when it is a global affair, global occurrence. An outbreak of disease that spreads quickly and affects many individuals at the same time and spreading from one person to person locally or globally. And it must affect, cover more than one continent, then that is called as pandemic. Now what is so this is epidemic, sorry, that is when it is moving to the one continent. And pandemic when it is going from one continent to another continent, and it is a much bigger affair, virtually gulping total this world population, then that is called as pandemic. Then comes the endemic. What is endemic? What happened that when the pandemic or epidemic comes, the human being start fighting against this. And our body also start fighting against this and gained immunity, natural immunity against those diseases. And after certain period, that particular disease will remain in the population but will not be that harmful. Then that particular disease is called as endemic. What exactly is the case in case of COVID? COVID, when it started, it was devastating. Hundreds and thousands of people dying every year, every day, every hour people are dying. But nowadays there is no dying because vaccines are now available, people have taken vaccine. Another thing is that our body also has gained some immunity out of this because of the exposure to COVID. So COVID slowly has become endemic. So this is Bangla যেটাকে বলে যে গৃহপালিত. COVID has become গৃহপালিত. Now it's a paid one. So it is not causing that harm. So that stage is called as endemic. But then what is syndemic? Syndemic is actually there when there is epidemic, those people who are already suffering from other diseases, they are the most affected one. Say for instance in case of COVID, those people who are suffering from diabetes, who are suffering from cancer, who are suffering from other diseases, asthma. I have seen many students, people, they died of COVID because they had asthma. Asthma created havoc at that time. Other problems also, rheumatoid arthritis and all these things, that is becoming manifold, severe. That is called as syndemic. So there are virtually four types of pandemic is there, epidemic is there. Pandemic, epidemic, endemic and syndemic. Now I am coming one by one that work that we have done at North Bengal University. First is COVID-19. As I told you that when COVID-19 broke out, we have seen that North Bengal is most susceptible because North Bengal, most of the part is very remote portion, particularly the hilly North Bengal, hilly regions of North Bengal, the Darjeeling Hills, Karshiang Hills, Kalimpong Hills, these thing regions, and virtually they did not have much medicinal exposure, doctors and other things. So how to cope up with this particular disease? So what we have thought that they are very much close to the nature and a lot of plants are there. Many plants are there in their backyard which are medicinal. Say for instance Chirota, Bashok, Tulsi, Ketu and all these things, plants are there. If we can fight with those plants and if we can succeed, that will be the best. Because that time no laboratory virtually was working or thinking seriously on using plant parts or plants to fight against COVID. So we have started working. Now before that, but there is not, because everybody knows nowadays, and I am just skipping these things, how the COVID looks like, how the anatomy of COVID and all these things. Infection, how infection, as you know that the genomic DNA is negative stranded RNA, and when it sits, just simply push the RNA molecule to our body, which is done in any of the virus, in any cases of viruses. They push and then they multiply and then burst the cell and human cell and come out, and again affect the another cell. This way, this infection goes. Yes, yes. Tell me. Huh? Mic. Mic, huh. Now you can hear properly? Oh, okay. But why we could not do these things? Why there is, so till date, against COVID, not much medicines are there. Why you could not produce medicine? That is because scientists and medicinal professionals are striving to fight possible therapeutic remedies, but they could not. There are many ways they have tried, but they failed. Vaccine is the only alternative, and vaccine works very good, but then there are so many population, 10 billion population, and giving 10 billion population 3 doses of COVID vaccine is not that easy. There are many African countries still dead, they did not have vaccine much. So this is the problem. So only thing is that only what we can do is to cope up with the available remedies, available resources that we have in our backyard. This is also, everybody's. And this is the way of the things. I am not going detail that how it is done, but through my talk, I will talk about these things. Now, when we have taken this in our laboratory, we thought that which plant we should take first. Then we thought that because the symptoms of COVID was very much similar to that of the cough and cold, normal cough and cold, but it is much more severe, and there is a breathing problem was there. So we thought that why we shouldn't start with our normal phytocompounds or phytomedicines which are present in our backyard, the plants which are present in our backyard. আমাদের উঠোনে যে প্ল্যান্ট পাওয়া যায় সেটা দিয়ে যদি আমরা প্রথম শুরু করি. So we have here cut few plants, say for instance Calodendrum infortunatum, that is Ghetu. Then Vashak, Tulsi, garlic, turmeric, clove and neem. These are the plants that we have earmarked to fight against COVID. This is the Calodendrum infortunatum or Ghetu. This plant we have started for the first time, and in first time itself it creates havoc, because from these plants we have taken, actually 11 species of Calodendrum we have taken. All the 11 species are not found in Bengal, but some of the species are found in remote part of Assam. And thanks to my friends and colleagues, they have collected these plants, those species of Calodendrum infortunatum from Assam and sent it to us, and then we have analyzed them. We have done the GCMS analysis and LCMS analysis, and then we have gone for bioinformatic analysis, because in COVID, there is no other way rather than bioinformatics, because the models are not there. There is not that you are making a model rat or mouse COVID positive and then you are adding the medicine. That is not possible, because COVID doesn't infect them. So you have to take only the bioinformatic way to do these things. So we have collected the phytocompounds, and altogether 68 phytocompounds we found from Calodendrum infortunatum. And then we thought, we analyzed the COVID genome, and we found there are 3 proteins only 3 proteins, which are, if we can stop these 3 proteins by plugging with phytocompounds, then the COVID it can be managed. These 3 proteins are spike protein that you see, the spike that is coming out of the COVID virus, main protease, which is actually causing the disease, and RDRP, DNA, RNA dependent or DNA RNA polymerase. RNA dependent RDRP. Because these things, the 3 proteins can be stopped, then the COVID infection and COVID devastation, COVID spreading can be stopped. So, out of these things, in a result in a nutshell, we found that from this phytocompound that is COVID Ghetu, that is infortunatum, from there we have, as I told you, that we have found 68 phytocompounds. Out of these 68 phytocompounds, we found 1 phytocompounds which is creating very good results, giving very good results. One is, and then we have done this analysis that is called as terazerol. Terazerol, sigma sterol also we have taken, and other compounds taken, but in all the cases, in all the 3 proteins, we found that terazerol is giving better results. Minus result is actually good. If the minus result is more, that means binding is far more better binding, and the plus result means less binding. Okay, so this is minus 7.5, minus 8.4, and minus 7.4. These things. Whereas that time there are some other allopathic medicines also there. Say for instance, favipiravir, remdesivir, and other things, arabidol, but they are not giving that good results. But our compound, that terazerol, is giving very good results. So we have published these things, and then we have seen that this is the only molecule which is creating problem. So we have recommended that terazerol or the G2 can be used effectively against the COVID. All the three proteins. This is the only effective. Only bioinformatics, because you cannot check it with the. We have started these things with yes. We have a collaboration with Chinese laboratory, and we have started. But that time, now when we have started that time, that was not there. It was very difficult to do these things. So that time we have done it only in bioinformatics, but our result have found right over there in when it's done in the laboratory, lung cell, other things, no, that we have done, and it is showing this. I'm not showing in detail the results, but I'm telling you that is there. Might be problem করছে না আচ্ছা এবার ঠিক আছে হ্যাঁ ওটা হাওয়াটা আসছিল আচ্ছা হ্যাঁ actually what happened that when I've started this work, I'm telling you a story. When I've started this work, that and when you got a very good result with G2 or Cladodendron infortunatum, then I have consulted the Ayurvedic books. And in the Ayurvedic book, I found a story which is very much, I found is fascinating or amazing. Because G2 is considered to be the husband of goddess Shitala. In Ayurveda it is said that G2 is the husband of goddess Shitala. And goddess Shitala is the all viruses, yes, particularly pox and other things, no? Viruses. So, yeah, G2 is also works in pox. So, only the G2 since the husband of goddess Shitala, so G2 can meant all these things. So, that's what I found. So, from G2, we have analyzed that terazerol is one of the molecule that is done, and then we have terazerol we have chemically synthesized also in the chemical laboratory with our chemistry department. We have synthesized and purified form, so that now it is can be given. But only problem with us, since I am in from botany department, I don't have a BSL 3 laboratories, and I don't have the permission to work on human being. I can do is lung cell work you can do, but on human, then I have to collaborate with the hospital. So we have thought of the things, but that time we could not do it. Anyway, so magic molecule terazerol, as Sat pointed out, that this is the molecule which works with all these three proteins. So one molecule which can unplug all these three proteins, and then reduce the effect of those proteins. The activity of those proteins can reduce terazerol. And we have published in JBSD. JBSD, this has become very popular because that was one of the pioneer paper on COVID, phytocompounds, COVID. Because this was published in 2020, that's why not very many paper was there. When we have got these things, then and there we have published these things. That time, when they send it to journal, they take only 15 days to publish it. Because there's a competition, all the other hot topic. That was very nice time for us, because whatever you are publishing, you are usually published, whatever you are saying. So that was there. Then after that, since I thought that we got a very good results in G2, we have taken other phytocompounds also. But as I told you, our rule was to take those phytocompounds which are used against cough and cold. Because COVID is more or less the similar kind of this COVID, cough and cold. So we have taken Justicia adatoda, that is Babashok, Osmum centum, that is tulsi, and Swadiya chirota. These things we have again done, and from there we have got 17 phytocompounds, and these 20 phytocompounds are also very good for interaction, all these things. So I am not going in detail because these are hardcore bioinformatics, but you can see that we are getting very good results. And there amyloidine and xanthine, all these things we have found, and all these three spike proteins are taken care of. Spike protein, MPRO, and RDRP. All these three proteins are taken care of. How it has targeted, and the interaction analysis I have gone, but these are hardcore bioinformatics, but I am not going in detail. If you want students, I can send my papers. In fact, we have made a booklet of these things, all my COVID papers that is published in our laboratory, from the laboratory, that we have given the name that COVID research at North Bengal University, this one. COVID research at North Bengal University. If you wish, just send me an email. I can send you the soft copy because I have only one hard copy. I can send you by email. And we have again published these things in JBST. Probably you have seen this paper also. After that time, there was a concept that repurposing of drugs. Repurposing means the drug which is used for some other diseases can be used for treating other diseases. Say for instance, chloroquine. Chloroquine is actually used against malaria. But that time, since there was no medicine, doctor used to prescribe chloroquine for COVID also. So that is called a repurposing of drugs. But chloroquine failed miserably. Chloroquine didn't work. Then at that time, as we got good results in phytocompounds, particularly the compounds which are present in Ayurvedic cough syrup, so I thought that why the allopathic cough syrups are, whether it works or not. Usually, I myself is actually a victim of the cough and cold. I see seriously and very frequently I used to have this cough and cold problem. So when I suffered from cough and cold, so doctor used to prescribe some cough syrup, and I have seen the ingredients, and there are some certain ingredients which in that case, the dexamethasone, penicillin, and dexamethasone. These three ingredients are very common. Penicillin and dexamethasone are steroid. It are all steroid. This penicillin and dexamethasone are steroid, but dexamethasone is not steroid. So what I thought that whether they will also be effective against COVID or not. So we have done extensive study with these three phytocompounds, the three compounds against the COVID. With all these three proteins, that three proteins I have shown. And then we have done repurpose. Will be repurposes. So whether if nothing is there, if cough syrup also you can take, you will be relieved. That's what our submission was there at that time, and it works. And we have done molecular dynamics up to simulation, up to 200 nanoseconds, 200 nanoseconds we have done, and we found that it's absolutely working well. RMSF value is there. How it works actually? See, when COVID comes, this is COVID molecules coming to our body and ultimately gather place in our lung. When it comes to lung, activation of the immune response. Our body starts immune responding. And immune system is actually not good to our body because it produces a lot of immune storm, cytokine storm. And cytokine storm actually make us weak, or diseased. So activation immune system and cytokine storm take place. So there will be fever and inflammation and cough. The cough reflux is there and cough is there. Sigma opioid receptor is there. So these things. Now what happened when these particular three drugs we are giving, dexamethasone, penicillin and dexamethasone, then what happened? They first stop the cough reflux. So there will be no cough. You will not be having any misery. Second thing is that it stops the producing of cytokine storm. When there will be no cytokine storm, then your lungs will be properly not affected. Only thing is that our lungs become fibrous due to the presence of due to cytokine storm. If you can stop cytokine storm, then it will be automatically cured. COVID cannot create much problem. So this is the thing that we have done, and this we have shown these things for the first time, that these three particular compound which are actually used for cough and cold, normal cough and cold, normal when you have simple cough, doctor used to prescribe, those things can be used effectively against COVID. And again we have published these things. Now the thing is, there is a catch over here. You see, we have published this in 20th August 2020. 20th August 2020. These are third, fourth publications. 2020 we have done these things. Yes. And then World Health Organization has given corticosteroid. They have also prescribed corticosteroid for fight against COVID on, you can see, 2nd September. Virtually, we are the first to prescribe this one at in our paper. Then corticosteroid was prescribed by World Health Organization. World Health didn't prescribe first because they did not know at that time. When we have published these things, then and then they have also prescribed that this can be used. Particularly penicillin can be used properly. That shows that they have seriously taken our discovery, what we have done at North Bengal University, they have taken it seriously. And then Science magazine also said that these particular compounds may be effective against COVID. How doctors treat COVID? Their doctors also started giving penicillin and other things. There we found afterwards that they are giving these things.
You use dexamethasone or prednisolone?
We use the combination of dexamethasone, prednisolone and dexamethasone, 3. Simple cough syrup combination. You pick up any cough syrup, we'll be having this steroid. Very mild steroid, but that steroids are there. So these things. So when we have discovered for the first time we have shown that these are effective, then first who is the prescribing? World Health Organization. Then Science magazine also says that doctors are using. In fact, in Bombay I have seen there is a big queue of buying penicillin at that time, because everybody is taking penicillin. And they have recognized us. They have sent a certificate saying these things, that your paper has given development goals, United States US Sustainable Development Goals. They have sent a certificate through Elsevier that congratulations on this is paper. Your paper has actually given these things. Now, second part of my work, my talk, infection issues. Usually, COVID is actually a very weak virus. It's not a very strong virus. 80% of the people used to be recovered as such. Simply you give them raw tea, they will sip the raw tea and they will be recovered. So 80% of the infection will go off. Major cases 50%, 15%. And severe cases where people used to die is 5% only. And all the death is due to this 5%. In most cases, maybe many of you up here who were suffering from COVID, but now they have become well. So that is that. But there is a underlying part of it. Normal infection 80%, you can apparently say that you are cured, you are fine, but you may not be fine. Maybe there is a hidden COVID problem in your body, and that may create problem. The classic case is the case of Dr. Palash Mondal. Palash suffered, one of my colleague, and I am very sorry for that. Dr. Palash suffered from COVID and become absolutely cured. He thought that he is cured, but it was there, and slowly his lung got affected. And when it was diagnosed that his lung is affected, 80% of his lung is actually non-functional, become non-functional. And within 2-3 days, he died. So post-COVID or long-haul COVID is another devastating problem. So need to be taken care of. So what we have done, the post-COVID syndrome and management. So we have done, after doing the COVID, we are seriously working on post-COVID syndrome because we have seen a lot of people are suffering from post-COVID. What are the possible sufferings? Sufferings are depression, bipolar disorders, stress disorder. Then other things are there. These are Alzheimer's disease are becoming a headache. All these things are there. So these are all the byproduct of COVID or the post-COVID symptoms. Particularly mood change. I have seen many people. One of my students become COVID affected and become cured. But then she started shouting on us, on me also. I understand she is not well because she is having problem with COVID. After that, no, that mood change is there. Suddenly mood change, suddenly become violent. So this was there. Now why it is happening? So we have done seriously the molecular level. We have studied the things and we found that a drug which is wonder that is called as cannabis. Cannabis usually consists of two phytocompounds. One is cannabidine, cannabidol, and a cannabivardine. These two phytocompounds we found, and we found that they are actually can cure the post-COVID syndromes to a large extent. How? First of all, you understand this? This is cannabis. This is cannabis sativa and cannabis indica. This common gaja jake bole. Gaja. So we have done the phytocompound analysis. We have done the GCMS analysis, GCMS and LCMS. So we have done the bioinformatic analysis, and then we found that it works. Now, how it works? I'm showing one by one. This is normal brain. Now, normal is affected with COVID. Your body is affected with COVID. So, brains usually have, our brain usually have receptors. Many receptors. Cannabinoid receptors also there. Even if you have other drugs, when the people used to take drugs, when they become intoxicated because the receptors are occupied with those drugs. Whenever there is a drug, no, you are taking drugs, the brain's receptors are affected with that. As a result, they don't work. That's why we don't stand properly, we talk nonsense, all these things happen. Now what happened? That these receptors are now occupied by COVID molecule. Then you treat the COVID is treated, then what happened? That body will become normal, normalcy will be shown, but body will not be totally normal. Because after 6 months to 1 year, the COVID molecules remain attached with our receptors, brain receptors. As a result, sometimes your mood is changed, sometimes there is tremendous headache, sometimes there is nausea, sometimes there is a bipolar syndromes are there. A person is watching TV, suddenly he started crying. These things are there. Stage by behavioral changes have taken place. So all these things happen. Now how cannabis works? As I told you that cannabis usually have two compounds, cannabidol and cannabivardine. Now the cannabidol and cannabivardine also binds with these receptors. Those receptors where the COVID molecule already has bound. When a person is affecting with COVID and taking cannabidol and cannabivardine, then what happened? The cannabidol and cannabivardine go to the brain, and since cannabidine and cannabivardine has a stronger bounder than that of the COVID molecule, so they will replace the COVID molecule, uprooted the COVID molecule and go and sit over there in our brain receptors. But their shelf life is only 2 to 3 hours. After 2 to 3 hours, naturally they'll become detached, and your brain system will become normalcy. That is shown that you have shown the normal brain affected by COVID. So this is a COVID affected brain. So COVID affected brain. Then we have added cannabidol and cannabivardine. So cannabidol and cannabivardine will replace the COVID molecule. COVID molecules are replaced. It is replaced, and then this cannabidol and cannabivardine also will be replaced, and the brain receptor will attain the normalcy. That we have shown, step by step we have shown, and this has again published. And this paper has become very popular because as this is cannabidine or gaja is there involved, and then This has become very popular in local newspaper also. They have done a big news on these things. That cannabis is most corona bish, theka theke dawai gaja. But when we have just stopped that writing that paper, we have written that paper, in the meantime another problem has come. That problem is COVID-associated mucormycosis or CAM. Suddenly it is found, because during COVID now, our immune system is terribly compromised. As a result, another fungus used to come and bind with our body, and they create problem. That is called as mucormycosis. And mucormycosis is, that is called as CAM or COVID-associated mucormycosis. This has become, and many people have died of these things. And not only that, even if you don't die, see the condition of the things, a part of the body has become rotten and you have to cut it out. So that is a very dangerous thing, very ugly affair. So we have taken another approach, but we have taken a different approach in this case. Here we have taken the reverse pharmacology or network pharmacology studies. So we have done these things and we found two molecules. There is network pharmacology, and we found that haldi, that is turmeric, and neem. These two molecules, two plants we found are very effective against mucormycosis, because this may boost the immune system as well as they stop affecting the effect of mucormycosis. Actually what happened, mucormycosis, when mucormycosis takes place, our blood vessels become thrombosis due to the presence of mucormycosis. As a result, blood vessel dies and that portion become blackened. And iron content in our blood become very high. না নিমের রস এখানে নিম এবং ইয়েকে ডাল এটাকে বেটে খাওয়ানো হয়েছে নিম এবং হলদিকে একসাথে বেটে বড়ির মতো করে খাই দাও and we have shown that this particular neem and turmeric has actually, this is the scheme that we have shown, and this scheme has been accepted in paper. And then we have studied some other work, because some of my collaborators are also working on these things. One paper has come from South Africa that they have done their one, they have found one algae which is we may cause may give protection against COVID, that we have published in very good journal Molecule. Our next was is on monkeypox. Since the COVID has done, the new virus has come, that is called the monkeypox virus. And monkeypox virus problem is still there. When the monkeypox virus has come, then one of my girl students from Tripura, she just told me that, sir, in Tripura, in the village region, when a baby is suffered with, or a person is suffered with chicken pox, then that person was allowed to lie on the leaves of phyllanthus, that is আমাদের যেটাকে বলে আমলকি আমলকির পাতার উপর তাদের শুয়ে দেওয়া হয় তাদের শুয়ে দেওয়া হয় I said that why amlaki? Then amlaki leaf must be having something. Then we have collected amlaki leaf and we have analyzed the GCMS analysis, HCMS analysis we have done, and then we found few compounds. Then what we have done is that this was the one. We have seen that how it works, and I am not going in detail because maybe I am already late. So these things, and we found that how amlaki actually works. This is the schematic diagram of how it works. These are the 3 proteins again, and the 3 proteins amlaki compounds have go and bind, so that it doesn't work. And this is the network pharmacology that we have done. This is a bit complex work. I am not going in detail of these things. This is the schematic diagram. This is the phytocompounds that from amlaki, that is Phyllanthus acidus, and then this is the way it can stop. And this is the paper. Another work we have done on monkeypox, that is the development of vaccines, because vaccine development is very important, and regularly you have to develop vaccines for these things. So that time we have developed the vaccines. We are one of the pioneer in the world to develop a vaccine against monkeypox, because usually people used to use smallpox vaccine against monkeypox, because work is similar. But monkeypox doesn't have a dedicated vaccine. We are the first time in North Bengal University to develop the monkeypox vaccine. This is the paper. Journal of King Saud University, Saudi Arabia, because of my collaborators was there, so they have published it. Then fight against viral diseases, Nipah virus. Recently we have done this work on Nipah virus. We have developed a vaccine against Nipah virus, and this has been published. This is the way we used to do these things. And what is vaccine? I'm not going in detail of these things. And there are various steps of the vaccines. You can read this thing. Sequence retrieval, epitope production, vaccine construction, secondary and tertiary structure prediction, validation, and molecular dynamic simulation, Excel analysis. If you can do these things, the vaccine will be produced, and we have produced vaccines. And this is the paper. In silico vaccine construct. But yes, we have talks are going on with the Odisha laboratory, Bhubaneswar laboratory. So they will produce real-time vaccine for us. I can't remember because one of my students is involved. In fact, one girl is there. Amrita was there. No, Amrita in North Bengal, Midnapur University. Amrita's laboratory, Amrita's institute. They have these things. One Shikhao something, no? Shikhao Anushandhan, that university, Shikhao Anushandhan. They are interested in doing this. Very good work actually. They have done a very good laboratory, and they said that, sir, we will make vaccine for your paper. So this is the thing, monkeypox vaccine and multi-epitope. These are the multi-epitope vaccines that we are doing. Another vaccine we have already prepared. Now it is communicated to a journal for possible publication. That is called as Oropochi virus. That hasn't come in India, but in South America, this has become havoc. And 1,500 people, odd people died. 500 odd people died already of these things. So we have already made vaccines out of these things, and we have given to the journals for publication. And also, we also will try to produce this vaccine. This is a nutshell. And the Bioinformatics Facility Center and Vishwabangla Genome Center, this is the building, and there are a lot of software which is there. If you happen to, the students, I'm talking about the students, if you happen to come to North Bengal, you can visit our laboratories. These laboratories are good, moderate facilities are there. You can visit over there. And actually, this is the only center in whole of North Bengal, which cater whole of North Bengal, part of Bangladesh, Bihar, part of Bihar, particularly Bhagalpur and all this region, and Nepal and Bhutan, and also Sikkim. All these things are catered with only this center, the Center for Bioinformatics at North Bengal University. We published sometimes, of course nowadays become irregular, the Bioinfoline, one newsletter on bioinformatics. And these are some of my students and collaborators that they are staying there. So this is Dr. Ashim Botra of Raiganj University. He recently died. I'm sorry for that. This is Dr. Indrani Sarkar, who is there in AIMS Rishikesh now. This is Professor Louis Tiza of University of New Hampshire, USA. Professor Philippe Normander of University of Lyon, France. And Professor Maher Jitari of University of Tunis, Tunisia. These are some of my collaborators. My other collaborators also there. I'm not adding this name. And these are some of my students, current students are there. And thank you.
Thank you, Professor Sen, for your illuminating lecture. The deliberation is now open for discussion. Any question from the students or the researchers? Before someone asks any question, let me ask some silly questions.
Thank you, sir.
First thing, you tried for vaccine development against Nipah virus and
Monkeypox virus.
why did you not try against COVID?
That time actually, I did not have much expertise on this. To be very honest, since the outcome of when COVID came, we felt the need of producing medicine,
Instead
particularly
of
vaccine,
vaccine.
instead of medicines. So that I have started learning how to produce vaccines, and then we have done the monkeypox. By the monkeypox, we have understood, but that was a monoepitopic vaccine. That was not very effective. And then afterwards, that we have done, that is multi-epitopic vaccine. And the last two ones that we have done the RNA vaccine, that is COVID also RNA vaccine was there, no? That same RNA vaccine we also made.
Same RNA,
Same type of RNA vaccines were made. So this is very robust vaccine. And now what I am thinking that nano vaccine, nanotechnology also can be used for producing vaccine. Maybe next paper or next our product will be on nano vaccine. Nano vaccine is much more effective.
My second question is really silly. এই গাঁজা নিয়ে কাজ করলেন যে পুলিশ ধরে নি
পুরোটা ইনসিলি কথা হাওয়ায় হয় কাজ
you do not have not to face any legal problem
no
cannabinoid আছে সে তো আছে but working with cannabis plant
actually তখন কোভিডের লোকের এত ব্যস্ত ছিল যে গাঁজা নিয়ে মাথা ঘামানোর সময় ছিল না
meanwhile any question from the students or research
yeah sure professor yeah
which is in the COVID virus actually.
Yeah.
Why you are not concentrating TLRs? If TLR is upregulated, then all virus will be blocked.
Yeah, that
in
is
the
that I
cell.
know,
Why-
but
why
that
you
time,
are not?
I do not have time to do these things because TLR working would be a very difficult one. You can agree with me. So I thought that the immediate solution is to work with these 3 viral proteins. That's why we have taken. But TLR can be taken.
any more question from anyone else
If you have questions afterwards, you can write it to me. My email ID is given there, so you can write it. Another thing is there, there is in my abstract book, you'll find there is a silly mistake that is typo, that will be outbreaks or something like this. There is a spelling mistake. Just correct it. Afterwards, I have seen that there is a correct spelling mistake. No.
okay thank you once again sir
Thank
and
you.
we are closing the day's final scientific session right now
Thank you, sir, for this wonderful session. Now it is my privilege to welcome Professor Ananta Kumar Ghosh, former professor, Department of Biotechnology, IIT Kharagpur. Our madams will be felicitating him. A big round of applause for sir. After half an hour, there will be cultural program. Thank you, sir. Now we will have a tea break for half an hour. After that, there will be a cultural program in open stage. So we will request all of you to join there. Thank you very much.